Good job, thank you.
I didn't know that.
Got it.
You
Did you? Someone started a recording?
Was it you?
I did.
Oh, thanks, Sandra. That's good, that's good.
So Tom, I know it wasn't this inept when you were here, but you can watch us in action.
There we go.
Good deal. Well, first of all, thank you everyone for joining us today. It's a pleasure to be able to get folks together and talk about Lawrence Bruner and to recognize all of the accomplishments of our students, current and former, and all that they've done. And we especially welcome Dr. Perring with us today. Thank you, Tom, for agreeing to participate in this, and congratulations on your selection. It's certainly well deserved. So
My honor, thank you.
And I also want to thank the Bruner Club folks for all the effort that they put into this to make this work. It's very exciting. Lawrence Bruner is an interesting guy, and it's fun to be able to talk about him a little bit. And I won't spend too much time. There's a lot that we could cover. There are boxes and boxes and boxes of Lawrence Bruner material in the archives that is yet to be opened up. So hopefully someday we can mess around with that a little bit. So Bruner Week is designed to recognize Lawrence Bruner and his contributions to really the growth of students in the Department of Entomology here at the University of Nebraska. This is just some bioinformation. He was born in 1856, so this would be his 166th birthday this year. He was born on March the 2nd. On this day, 166 years ago, his mother was probably very anxiously looking forward to delivering a child. But Bruner was well known for loving students. He had a real passion for training students and engaging with students. This is a great picture of the lab that was here on East Campus a long time ago. This was back in the early 1900s. And Lawrence Bruner is sitting right over here. He's got the blue arrow pointing at his head. He's watching all of his students. So Tom, interestingly enough, just in front of Bruner, seated right here, studiously looking at something with his microscope next to him is Harry Scott Smith.
Oh, wow.
Who is an icon in biological control and was a student of Lawrence Bruner's, and actually married Lawrence Bruner's daughter, before he eventually went to California and had a tremendous impact on biological control. That's just one of many students that Bruner trained, and I just wanted to give some comments that students made about him and his mentorship. This is from Myron Swank. Myron stayed here in the department, became department head after Lawrence Bruner, and he wrote a lengthy obituary about Bruner, and he and Bruner had a special relationship. And this is just from his the obituary. He says, the 43-year-old man, that's referring to Bruner at that time, if you do the math, that's 1899, of much superior knowledge and experience about birds. Bruner loved birds and insects. Swank loved birds initially and eventually switched to insects. And on a busy day, gave up a large part of his afternoon telling the 16-year-old lad, Swank, about his recent trip to South America, showing him the specimens of bird skins and eggs brought back, as well as the insects collected, giving him a copy of the 1896 report, which was a big slab, and encouraging him to enter the university upon graduation from the Beatrice High School. That is what actually subsequently took place in the fall of 1901. So Swank ended up coming here to the University of Nebraska. And he shared this about Bruner, that it may seem odd to some readers of this sketch of Professor Bruner's life and work that Swank should make so much mention of the several boys who assisted him from time to time in his labors. But any one of them can testify, as now does Swank, how their interests and work were intertwined and guided by those of Professor Bruner himself, so that in a large sense, their activities during these years became also his interests. I like that at the end. It's kind of a nice touch of what makes a mentor effective. It's not just a one-way thing. So Bruner was engaging them in his work, but also he was engaging in their work, and he was learning and growing as a result of it. So it was a great mentor. This is a picture of Bruner and some of his boys, and I'm not sure who the lady is over here. Marilyn's not here, so she can't fix me on that and tell me who it is. But this is Bruner seated in the middle. He's got his basket, and you can see they're all dressed for field work in their suits and ties. And Myron Swink is standing here from our perspective to the right of Bruner. And he's got his binoculars. They've all got their binoculars. They're out on a bird/insect collecting trip. But you'll also, if you look back at the old pictures, women were also very active in entomology here, which is very interesting. This is Clarence Mickle. So Clarence was another one of Bruner's early students, and he went on with a career at the University of Minnesota. He said, my recollection of Professor Bruner is of a kindly patient man with an absorbing interest in entomology, which he somehow communicated to his students. We see that with a lot of our faculty now. His lecture notes, research notes, and often his first draft of research manuscripts were written on the backs of envelopes, which he systematically saved for the purpose. I remember his lectures as stimulative and informative. He was a good field collector, and I learned considerable entomology from him while on collecting trips with him in the field. So I've got we've got an envelope here that Ken Pruis donated to the department.
Wow.
This is a letter. Sorry, you can't see it here. Let me see if I can swing it over. Let's see here. All right, so this is a letter, and you can see up in the corner there is a grasshopper that he drew. So I'll pass this around the room here so that you all can kind of look at one of his envelope works. So it's kind of cool.
Wow.
Yes, I don't use envelopes. That's what Post-it notes were invented for. Right? And another student, Dwight Pierce, who's another prominent IPM guy. He said the professor was a short, broad-set man with a pompadour hairstyle. He spoke with a soft voice and a quaint sense of humor and had many interesting little stories about insects. As a boy, he was entranced by grasshoppers, and when supposed to be plowing, he was often found lying on the ground, watching at close vision the intimate activities of the wonderful grasshoppers. So he grew up this way, and he had a large collection of bird bodies and insects that he kept in a barn at his house when he was a boy in West Point, Nebraska. This is one of my favorite pictures of Bruner. This is Bruner here on the left. I don't know what was going on in this picture, but if you look at old pictures, people typically have very somber expressions because they had to hold still for quite a while for a picture to be taken. This particular picture is interesting to me because somebody said something or did something, but Bruner's got a nice grin on his face. He's being entertained by something. It's kind of fun to see him smiling. And this is a nice reminder that he had a great sense of humor, and there were interesting little things that he said and did where you could see his wit. And one of those, this is a little bit lengthy, so bear with me, but in 1915, the Panama Pacific International Exposition, which was an international fair, World's Fair that was in San Francisco that year, they asked every state in the United States to send their most distinguished citizen to that exposition. So there was a lot of debate in Nebraska about who they were going to send. The front runner was William Jennings Bryan, if you know who William Jennings Bryan is. He was a very famous politician and orator. He ran for president of the United States a couple of times unsuccessfully. He became the Secretary of State and is probably best known for being the prosecutor in the Stokes monkey trial, in which he argued that evolution should not be taught in schools. So he was a very, very prominent citizen. So this is from the Lincoln Journal-Star about that particular event. This is from July 9th, 1915. So the committee met and each member recommended a different candidate, but after 3 ballots, Bruner was unanimously chosen. Committee member and Bruner nominator W.H. Desch from nearby Ithaca, Nebraska, which is by Meade, right, explained, this is a great agricultural state and Professor Bruner has rendered invaluable service to the farmers and the state in general by helping to free the plant life of Nebraska from insect pests. Bruner was at work in his laboratory on the UNL campus when a phone call came asking him to immediately go to the governor's office. He was unaware of the purpose for the visit, but he went. While waiting for Bruner to arrive, the committee announced their decision to Nebraska Governor Moorhead. According to the newspaper article, Governor Moorhead appeared somewhat surprised when told of the selection and asked, "Who's he?" Later, when Professor Bruner arrived, the governor asked him what his name was. Obviously, the governor wasn't quite sure what was going on here and was unimpressed by the selection. But Bruner was very gracious, regardless. Said in a mild and assuming manner, Professor Bruner, when called upon, thanked the committee for the honor that they had conferred upon him. He said that the selection was a great surprise to him, but that he appreciated the honor and would do his best to satisfactorily represent the state. Shortly afterwards, the governor asked Bruner for a biography, and Bruner said that he could probably find what he wanted in Who's Who. So, kind of, I thought it was well played. You don't. Everybody else knows who I am. Go there and look it up in Who's Who. So I thought that was very nice. The article further says that the selection came as a distinct surprise to many, inasmuch as the man selected has never taken an active part in politics and has chosen to remain closely engaged in his work rather than to appear before the public. So he seems to be rather low key, but he had a good sense of humor. And one other one I wanted to share, this is from an article that he wrote called The Housefly in 1908. And this is how he starts the article. It says, "Of what use are flies anyway?" This and similar questions have frequently been asked of the writer by persons who seem to think that each and every living thing has been created solely for some special benefit to man. And I thought, but that's a great comment. Most of us have had to deal with these kinds of questions as entomologists. So it's been going on for at least 114 years. Right? But Bruner seems like he really was very patient, worked as a great mentor with students, and he put a lot of effort into building this department and building this university as one of the few faculty members early in the history of this institution. And he was the whole Department of Entomology for a number of years before his students began to come into the department as faculty and to build it and grow from there. So his impact on the department here, his impact on the science of entomology, and his impact on the growth of the students that he trained here personally as well as professionally has been pretty profound. And I think it's well worthwhile to stop and look back at some of these people who have really made things possible for us to be where we are today and to benefit from the resources that are here and to grow and develop. And it's great to be able to hear Dr. Perring share his experiences now of how he came up through the department, how his career has grown, and to reflect back on these people that created those opportunities for him as well going forward. So with that, we'll wish happy 166th birthday to Lawrence Bruner, and I will turn the time over to Courtney Brummel. So thank you very much.
Good job.
Thank you.
Great. Hey, I just want to, can we give it up again for John for putting that together? Good stuff. I specifically was like, he's going to be good with the history stuff because I'm like, oh, I didn't know that. And of course, finding like the cool artifacts along with it, you can't beat that. So first of all, thank you everyone for joining. Thank you, Dr. Perring, for taking time for today and being able to also speak afterwards. So we really appreciate that. A couple just quick housekeeping stuff. So obviously we're celebrating Bruner Week, which is a lot of focus on the students, but also just the department. So there's some really great events coming up. And I'm sure everyone's being bombarded with emails regarding that. So thank you for your patience with that. We just want to make sure we're communicating and not leaving anybody out, since this is for in-person resident students and also online. So that's why a lot of our efforts are hybrid or communication going over email. So the big thing is tomorrow we have a huge networking event. So that's at 6 p.m. Central Standard Time. And that is specifically only over Zoom because we have 10 amazing professionals that are going to be at this event. And you know what, that's just a benefit of Zoom. So we gotta hunt the good stuff with the global pandemic, and Zoom can be one of those. So we're able to connect with people all over the country. So we're really grateful for that. If you're planning on attending and you haven't already let Ina know, definitely let her know, because she is really setting up a very intentional layout so everybody gets a chance to meet with each of the professionals. So yeah, just a little housekeeping on that. So thanks again, Ina, and Gabby and Rogan. I mean, all these individuals that have really made this week possible. So with that, I would like to introduce Dr. Thomas Perring as the 2022 Distinguished Alumni Award recipient. So we do have a plaque for him, and it will be getting mailed to him. So here's it just virtually to you right now. Yes, that's right. Thank you. Thanks for being so gracious with us. That's ridiculous. But we're really glad to have him back, actually, to the entomology department today for his special seminar on his entomology career path. So Dr. Perring got his, I'm just going to give you a little run through, but he's going to go more in depth, which we're excited to hear about today. But he got his start in entomology, working on his bachelor's degree at Oklahoma State University. He completed his master's in entomology at Texas Tech University, and then his Ph.D. from the great University of Nebraska. And that was back in 1983, right? So we got him back. He currently actually works at the University of California, Riverside Department of Entomology, which I'm assuming is where you're at now. Yes, right? So why he's joining us over Zoom again. Grateful for Zoom so you can join us from California. And he's serving as the vice chairman there. So Dr. Perring's research group just throughout the years has focused on a variety of topics. The main thing just being the overall development of environmental and economically sustainable pest management programs for insects such as mites that are threats to the agricultural crops, particularly in desert ecosystems. So subsets of this research effort have been studies on the epidemiology and management of arthropod vectors, and also the use of natural enemies in pest management. So this research that Dr. Perring has conducted has resulted in over 153 reviewed journal articles, which is blowing my mind because I'm still just working towards one. So that's amazing.
That just means I've been here a long time.
Well, that's why we're excited to have you here and share some of your wisdom. Also, he's been publishing book chapters, review articles, technical publications, and symposium proceedings, and 5 edited books. Dr. Perring has been active in a variety of organizations, and one that we're all familiar with is the Entomological Society of America, and he's been part of that since 1978. So, wow. So with that, truly, it is an honor to present Dr. Thomas Perring with the 2022 Distinguished Alumni Award, and we look forward to hearing more about your entomology career journey. With that, we are going to mute ourselves on our end, and thank you so much for being here.
Thank you very much. Thank you very much. It's really a pleasure to be here with you. I feel like you're all sitting in my office now here in Riverside, so just transport yourself out here. I'm going to go ahead and try to share my screen, and we're just going to get started on this presentation. So let's see if I can do this, and we're going to go to here. And everybody should be seeing my screen, right?
Yes.
Yep.
Something about dates.
It is, yeah.
So the biggest joke in my lab is we're always in search for a good date, you know. Well, it's really an honor being here. I remember fondly my time in Lincoln and being in the Bruner Club and chairing, being the president, chair, whatever we called it back then, of the Bruner Club. And so it's really nice to be back. And so today I'm going to start a little bit with where I've come from, because I always find when I talk with students, it's kind of like, well, how did you get where you're going, where you are? How did you, what's kind of a little bit about your history? And then I'm going to move into the integrated pest management in California dates. And I've got a subtitle here that says, pay attention to the little things. And hopefully that'll make some sense as we go through this presentation. But again, it's really a pleasure to be here. I see a lot of familiar faces in the Zoom. It's really nice to see all you folks again. Let's see if I can advance here. Okay. Well, I'm going to just start out. I started out of high school in 1974. I was born in Enid, Oklahoma, and some of my siblings, I have 8 sisters and 1 brother, and some of them are on the Zoom here. Yeah, that was 8 sisters and 1 brother, and we are all just a happy big family. I went over to Stillwater, Oklahoma, 45, 50 miles from Enid. And when I was growing up, like in many of the people that grew up in farming type communities, I like to hunt and fish, and so I wanted to go over to Stillwater and study wildlife ecology and in research because I could imagine myself with a up in the mountains with Yule gibbons and chewing on pine nuts and chasing deer around and all that kind of stuff. And so I went to Oklahoma State and actually while I was there, the wildlife ecology is in the College of the Sciences, but in the agricultural college is where entomology was seated. And I worked for Ray Eikenberry. I had a work study job when I started out as a freshman working in the entomology lab. And I worked on pecan weevil and some of the entomopathogens that were important in that. And by the time I finished here, I did go ahead and graduate with my wildlife degree, wildlife ecology and research degree. But there were no jobs available. So Ray knew a fellow down in Lubbock, Texas. And so I took the big leap from out of Oklahoma into Texas, went down to Lubbock and studied at Texas Tech University. But I didn't spend a lot of time at Tech because my research was out at the Texas A&M Agricultural Research Center in Lubbock. And I was kind of a guinea pig graduate student in that I was the first Texas Tech student in entomology to get my degree. But I had a major professor, Tom Archer, at the experiment station and a major professor, Dan Bartel, at Texas Tech. So I finished that degree, and well, that work was really interesting. I worked on grain sorghum in the high plains, and I worked on this critter. This is the Banks grass mite, Ligonicus pratensis. Great picture by Julie Peterson, which is who's on the faculty there. So thank you, Julie, for that. And this project was looking at host plant resistance of sorghum to the spider mite. And I spent a couple of years there. And in 1980, Tom Archer knew this guy named Tom Holzer. I guess it's all the Tom here. And in 1980, I took the leap up to Lincoln, Nebraska. And I lived in this building, which you now call Entomology Hall. It was a plant industries building, and my office was in the far top floor on the far northwest corner, where the wind would sweep down the plains and we would pretty much freeze up in that part of the building. Probably still the same now, but I was up there. Ken Purris's office was right around. ZB Mayo was right across the hall to the west. Sorry, I was on the northeast side, and ZB Mayo was over on the west side. Ken Purris would smoke his pipe in there, and it was awesome being up in that office up there. So anyway, I was happy and blessed to be mentored by this group of gentlemen. Tom Holzer's up at the top, and this is a picture he sent me just a few days ago. ZB Mayo and Jack Campbell. Jack was out at North Platte, and I did my research out there. And this other fellow here on the right is John Norman, and he was over in the agronomy department. And my project there was to work on corn up in the Sandhills, and I think there's still maybe corn up in the Sandhills now. Talking to Jeff Bradshaw and people out in the Northwest. And I worked on the Banks grass mite in that. So it's like, well, you know that animal pretty well. Let's just keep going. And I was reminded when I found this picture online by Jim Kalish that Jim and I spent a lot of time together. And Jim, I guess, retired a few years ago. But we spent a lot of time together in corn and counting spider mites in Nebraska, and Jim was just such a beautiful resource for photography and graphics and stuff, and we have some papers together from those very early days. This project was very interesting in that we were looking at the microclimate of the corn leaf as impacted by irrigation. So the idea was that if you can do irrigation management to mitigate some of the impact of these spider mites, and that if the plant went under stress, the leaves would warm up because they weren't transpiring. That microclimate would warm up, and you would accelerate the mite damage in that situation. So I was there 3 years doing my PhD, and then I really took the big leap coming out of the high plains, and I came into Riverside, California, where I joined a pretty motley group of folks that A lot of transplants from all over the U.S. And when I got to California, there was a couple of things that struck me uh, that were really important, that sort of reminded me that I wasn't in Kansas anymore or Oklahoma, that I was in a very different place. There are about 40 million people live in California, so it's a very crowded place. I'm about 60 miles east of Los Angeles, and there's a lot of traffic and a lot of cars. We have about 400 different commodities, agricultural commodities that we grow for a value worth about $49 billion. Those are huge numbers. But the other thing about California is that it's a very expansive state. It runs from about middle of Texas all the way up to the northern Nebraska. So it covers a lot of latitude. And so you can basically grow crops in here, starting down in the desert next to Mexico, come into Riverside, go up into Central Valley, and even higher. You can grow crops all year round. So when I remember doing my Ph.D. work in Nebraska, and you would plan, and then you had about 3 months to do your field research. And then you could come in and analyze the data and plan for the next year and see the results, that kind of stuff. That doesn't happen in California in working in agricultural commodities. And that you can be in the field all year. So there's advantages and there are disadvantages to that kind of thing. The other thing about California that's important is, on average, we get about 9 new invasive species per year. And this is since data has been collected back in 1956. We get one about every 40 days. About 3 of these actually establish as pests per year, and it gives us an economic loss of about $3 billion. So it's a big deal. I mean, these invasive species, California has an ocean border. It's got northern, southern borders, northern, southern, and eastern borders. And so we get a lot of new pests. And it turns out that in my career, I've worked on a lot of those. In fact, all but one are invasive pests. And this is I was just kind of going through my CV recently, and I just sort of put down, I've worked on 22 crops, 19 different pests. Again, all but one of those is an invasive species, 9 different diseases. I've developed IPM strategies, 16 different types of IPM strategies, and I've been involved in research with 21 different biological control agents. So the point of all this is that California, being in California, has afforded me an opportunity to be very diverse in what I've done over my career, which is now in its 38th year at the University of Nebraska, the University of California, Riverside. Well, today I'm going to talk to you about a pretty bizarre crop. This is something that doesn't grow in very many places in the United States. In fact, it grows sort of in the southern desert of California, a little bit in Arizona, and these are dates. Now, dates are an interesting crop. They take a tremendous amount of energy. These are palm trees, and they grow very tall. They have to be managed about 7 times per year. In January, you go and clean up the fronds. You have to dethorn it because the date as a desert adapted plant has got these real nasty spines. And if you don't dethorn, then you have to be working in those spines. And these things will go right through your boot, if even leather boots. And so, and anybody who's ever worked in dates have been probed by a date spine. So you have to go in and cut those thorns off of there. And so a group called the Palmeros, which is a handed down tradition from family to family, can climb these trees and dethorn and do this in January. Well, in February, out of the center of the crown is the spathe comes out and opens up into the flowers. And there are male flowers and female flowers on different trees. These are dioecious trees. And you harvest the pollen. Turns out that the male spathe comes out a little bit earlier than the female. You cut these pollen branches off, shake all the pollen off, collect it, and literally go then when the female flowers come out, literally go in and you puff the pollen onto the flowers, onto the female flowers. You then put a bag over the end of them to keep them protected, keep that pollen inside. It enhances the pollination. And then what happens in April and May? Turns out that the plant, because of this pollination, they actually set too many fruit for the plant to actually mature. So they have to go back up into the trees, and you have to kind of thin out the bunches a little bit, and so that the dates will grow to a good size for market. So they'll cut out the centers, and then here's the date bunch. It'll set on, it's about 25 to 40 pounds of dates. And in order to keep that branch from breaking, they have to tie it up to one of the fronds. Okay, so I'm telling you all this to give you an indication of how labor intensive this crop is. Okay, in June, grapes grow really fast. They develop into a larger size. The bunches get heavier. You can see these dates are nicely thinned out, really producing well. This is a picture over here of the trees that have dates set on them. It's just beautiful. Well, in mid-June, still the plant can't produce as many fruit as it set, and it goes through a natural abscission process. We call it the June drop. So a lot of these green dates and some brown dates, they fall to the ground. Well, in July, some of these brown dates, these upsized dates, actually have gotten stuck up in the bunches. Pardon me. And these become development sites for this insect called the carob moth, which I'm going to talk about in just a minute. In July, the growers go up. You can see some of the bunches, the dates are starting to turn color. They're going to start turning from green to auburn to brown. And they'll grow, the date farmers will go up and they'll either put a paper wrap around it like this or a mesh bag like this. This does two things. It helps to reduce a little bit of the insects, especially if they're the mesh bag over here. But this keeps the rain off of them. One thing that happens when dates get wet is that they'll spot the date and it makes them unmarketable. In August and September, this is a beautiful shot of the date ranch, and you can see the dates are ripening up. In September through November, palmeros are up in the trees again. They've got to cut all these bunches off and lower them down to workers on the ground. When I started this process back in working with dates back in the early 1990s, this is how they went up into the trees with these 60-foot extension ladders. Again, passed down from generation to generation, these palmeros working these trees. Then they went to lifts, and now they actually have these lift buckets, these lifts that go up, and they can harvest one tree at a time with these types of lifts that they have. Their dates are harvested, they're sorted and packed, and then moved to market. Well, one of the things that harvesting does is it puts a lot of waste dates on the ground. And you can see down here, these dates on the ground. Well, these become this overwintering sites for this insect called the carob moth. And this is the carob moth, actually, this is a female. And what she is doing is she is actually releasing pheromone and fanning that pheromone away. But what she will do is she lays her eggs with her ovipositor deep inside these dates that are on the ground. Okay? Those dates then will develop, the moths will develop very slowly in those dates. And then when it comes to the June drop, it's just at the right time for the emergence of moths from those dates on the ground. And then they will actually infest these June drop dates that are stuck up in the tree. Okay? And when she infests, again, she inserts her ovipositor deep inside the date, and here's a couple of her eggs. They will hatch. We'll go through maybe 3 or 4 generations. This is what the larvae looks like inside the date. Lots of frash, they become a fairly large larvae. These are pyralid moths. They go through 5 larval instars per year, and then they come out in these generations so that it really infests the harvestable date. Now there are a number of natural enemies that exist in our system. There's some trichogrammatid egg parasitoids. There's some larval parasitoids. This is Habobrachyum hebeter is one of the more prominent ones that are out there. There's even some pupal parasitoids in the family Braconidae. There are some ant predators that are out there, a couple of different, there are fire ant and some of the California harvester ants. And you can see this ant here literally picking up a larvae off of the ground and carrying it back to its nest. Now, when I started this process, the way that they dealt with this is they used malathion dust. Again, this had been going on. This carob moth was introduced into the valley, identified in 1982. And from 1982, when this picture was taken in the mid-90s, 1990s, these are dusting with malathion dust. Malathion is a pretty safe toxic into humans, but this is getting pretty ridiculous. And of course, when you get these tall trees, you got to have this ability to blow this dust up into the date bunches up in these trees. And they would do this about 4 times a year. Here are the really tall trees. I mean, you can just see the malathion dust raining down on everything. It got all over the dates. This is the powder. It got all over the ground. And what happened, of course, is it killed off all of our beneficials. This is an ant den after the malathion treatment, and you can just see the ants that have died because of this. Well, that basically was causing the negative impact of what excessive pesticide use would do before we really got into developing pest management projects. So when I came into this system and started working on this, first thing we realized we had to get away from the malathion dust, because that was just blowing away everything that we had in terms of any kind of pest management strategies that we had. So one of the things that had been done a few years before is that some researchers, Harry Shorey was one of those researchers, and Tom Baker and Jocelyn Miller here in the Department of Entomology, those fellows were all here. Jocelyn is still here, was looking at a pheromone-based strategy. And the idea was that we could use mating disruption, which was just coming onto the scene there, hadn't been around for very long. And the idea here was that if we could extract the pheromone from these females and identify what those particular compounds were that the males are responding to. This is just an old picture of those of you who know gas chromatograph, electroantennogram graphs. This graph shows when you can excite the antennae of the male, then you can identify which components are exciting those particular volatiles. And the components of this pheromones were identified. Then we teamed up with a company here in Riverside, Iskatech Company. It's run by Dr. Agenor Mafraneto, who was a postdoc here for a while, but he went off to run this company. And Agenor's company could actually produce a synthetic mimic of the pheromone of carob moth. It's very expensive to produce it. And when I came on the scene, we were just testing these blends out, getting them into the field. But this was the first major large-scale production. This is about 1,000 kilograms. And to produce this in the mid-1990s, early 1990s, cost us about $40,000. So these, 10 bottles that we see here, 12 bottles, about $40,000. So we made a joke then too about $40,000 a kilo. Sounded like we were running drugs at the time. But again, this is very expensive. And one of the problems with this pheromone is it wasn't very stable. It wasn't stable when you get out in the field. It would flash off, volatilize very quickly, and it wasn't very environmentally UV stable. So we had this issue of stability, and we also had this issue of this is our date palm with the dates up here. This is where the carob moth are doing their damage. And we had this problem about how do we get this pheromone, how do we stabilize it, but also how do we get it up into the trees? So a bunch of us got together and we decided that, well, one of the things that Iskatech did is they could put this pheromone in this kind of matrix. It's kind of a toothpaste consistency matrix. We still had that, and that would help stabilize it, but we still had to get it up into those tall trees. So one of the ideas that came up is, well, what if we loaded it into paintballs? And we actually shot this up into the trees. And so this was one of the best summers my graduate students and undergrads ever had, in that we set them out into the date ranch with paintball guns, loaded with these pheromone paintballs, shot it up into the tree, and it would kind of hit the trees and splatter out. And one of the ways you test the effectiveness of this, so the idea here, for those of you who don't know this mating disruption strategy, the idea is that you basically put enough sources of the pheromone in the environment that you saturate the environment with the smell. And when you do that, then when the female calls, there's so much saturated smell that the males can't find the females. Okay? So they also do their mating all at night. So this is all happening in the dark, so there's no visual cues that the males are using. They're just using the smells of the female. So the idea is if we took these paintballs and we shot these paintballs up into the tree, and we did that to every tree, maybe in a couple of places, we can inundate the date ranch with the smell. The way you test the effectiveness is then you essentially make a lure that's got the pheromone blend, and this is the lure. You put a small amount of that blend, and this is in a sticky trap. And if the pheromone disruption strategy works, the males will not be able to find this lure. So you put these traps out there, and you see, can the males find them? And so this is a trap that's got a lot of males that found it. This is another trap that has fewer males in it. That's our lure in the middle on these little septa. And so again, if the mating disruption is working, these traps are going to be empty or have very few males. Well, here's a graph that shows the data from our study. These are the average numbers of males caught in our traps in the field. This gray bar is in a block that has the malathion dust. The pheromone is here in the clear bar, and you can see across the field, and this is over the season of collecting, you can see that certainly the malathion dust, where we did not have any pheromone, still had a lot of males caught in it. So the malathion wasn't doing really the job that that the farmers are expecting it to do. The pheromone, on the other hand, kept the numbers pretty low. However, what happened is when we would treat, this is a paintball treatment at this time, we knocked out all the insects there, but then we'd start seeing them starting to creep back up. So this is in the pheromone plots. So then we thought we'd have to treat again. Pretty good, knocked them down, numbers start coming back up, treat again. Numbers are knocked down until they started coming back up, treat it again. You get the picture, is that it would knock the males down, but then it wouldn't last very long. So this is what happened on the infestation. So this is our control plots. What this tells us is that the pheromone was a little bit higher at 5% than the malathion dust. So we were protecting the dates, but none of these are statistically different. And the idea is that, well, the pheromone is at least giving us as good a protection as what the malathion's doing. Neither one of them are doing very much over the controls, but we're in the ballpark. Again, these were very early treatments of this particular pheromone stuff. So the results were acceptable, but it was hugely expensive, especially when we were treating about every 3 weeks. These paintballs are about $5 a piece. So to run this little trial was, I don't know, $350,000, which is way more than, which is about it on an acre's basis, which is way more than the $250 they were using for malathion. So anyway, point is that it was very expensive. So we had to go back to the drawing board and say, okay, this strategy looks like it could work, but we've got to reformulate it. So we went back to the drawing board, put it out in a different kind of a format so that it could be put into a caulking gun and actually applied with a caulk gun. And when we put this out, it would create this dollop. It's kind of looked like a Hershey's kiss on the plant. And this did two things. Number one, it didn't splat very flat so that the pheromone would flash off. In fact, it gave us a nice architecture. The formulation of this material was a little different than the original formulation so that the pheromone was even more protected. But in that architecture, pheromone would literally be released off of this in a very time-release fashion. And when we tested this, it gave us about 16 weeks of control or attraction to the moss. So we knew we could get a longer-term strategy. Well, so here's what it looked like applied to the palm fronds. This is the splat, or this is the dollop with the caulking gun. This is the splat. And we put this out then in a very large 60-acre date ranch, randomized complete block design. And here we are actually applying the splat. The palmeros were climbing the trees and put the splat out for us. A very huge project. These are the numbers of males that we caught in the traps. And once again, one treatment out here. And if we take a look then, these are the numbers of males in the non-treated. This is the malathion plots. And down here on the bottom, these are the pheromone treated plots. Okay, very good control. In other words, the mating disruption is working. The males cannot find the females in our pheromone treated plots. Now this is all great, but the proof is really in the pudding of what the yields look like. In our non-treated, the yields were, the infest was about 14%, 10% in malathion, and about 8% with our pheromone treated. Pheromone alone, no insecticides. So from these data, this is one of 2 years. The second years looked basically the same. And from these data, we were able to go to the EPA and the California Department of Pesticide Regulation to actually market and register this material. It's now called Splat EC, and it's registered for organic production, which is really awesome because farmers, as you know, at the time, organic was not big back in the mid 90s. But this work was being done in the mid 2010, 2014, 15. And you can see that, well, organic production is becoming more and more important now. So having this material for organic is good. Now, I bring that up, but also want to say that at the same time, we were still moving on with some other studies, some insecticide studies, because we had to get away from the malathion dust. And we identified a couple of materials that have now been registered. So the farmers who are in conventional agriculture, spraying insecticides, can use these materials. Malathion dust is not used anymore. And the organic farmers can use the splat mating disruption. Or farmers can actually use both, and some of them do. Okay, the second pest, very interesting pest, which is called the pink hibiscus mealybug. It's called pink hibiscus mealybug because it develops on hibiscus. It's pink in color, and it's mealybugs are these these heteropteran sternorincans that feed on the phloem of the plant. They extract out this sugary phloem. They also have this waxy exudate that comes out of glands on their abdomen. And that sticky exudate, their frass and that sticky stuff, it all gets all mealed up in what we call this mealy mass. So mealybugs are really nasty insects in terms of the mess that they cause, lots of honeydew production, lots of sooty mold, and lots of this mealy mass that we have. So this is the second pest I'm going to talk about. And it's got a whole different set of problems that are created by it, and a whole different set of solutions that I'm going to tell you about of how we have gone about working on this particular pest. Well, pink hibiscus mealybug feeds on a lot of plants. It's very prolific. It's 330 different species. This is hibiscus, it's the one that it's named after, and it can literally kill this plant with so many mealybugs that get on there. Now, we found out about this in the fall of 2014 when one of the date ranchers called me to come down and look at some dates that he had harvested that were coming to his packing shed. And they had these mealy white dates in there. So obviously this is not going to be able to be marketed. And so I started thinking, I asked him, so where do these dates come from? Because these packers receive dates from all over because clearly we've got a big problem in our date ranches. And he said had these 30 bins, it's about 30,000 pounds of dates that were all infested. And they actually got harvested. I want you to take a look at this picture from these date palms that were growing in the city of Indian Wells. Now, Indian Wells is in the Coachella Valley. It's just a little ways from Palm Springs. There are 228 golf courses. PGA Tour is out here. This is a very affluential area. But they also use date palms as ornamental trees. Now, these palms were actually part of an old date ranch. And when they developed this urban area, they left some of these trees for ornamental trees. Now, in other places, they literally will dig the date palms and move them. if any of you have ever been to Disneyland, you'll go down Disney Way and it's lined with these beautiful palm trees. All of those date palms came from the Coachella Valley date ranches, and they were dug out of the ranches and they were moved for landscape plants. These plants, however, were there from an old date ranch. And those date palms, those ones that were in the bins that I just showed you, came from these trees. So these date palms can actually be farmed, and it turns out that a farmer actually farmed these trees and harvested dates from these, and that's where these dates came from that were so infested with the pink hibiscus mealybug. Well, in a sense, that was good news for us because there is a separation between the urban area and the agricultural area in this Coachella Valley. So the problem, at least at this time, was in the urban area. Well, this is what I was just talking about. So this is a map of the Coachella Valley. Palm Springs is up here, Cathedral City, Indian Wells, La Quinta, all these places that if you're going to come out and in the wintertime and vacation in Southern California, a lot of people go to this area. And this out here then on the green, this is the agricultural area. So the agricultural area used to occupy about 99,000 acres. The big commodities are dates, grapes, and citrus. They also do have a lot of vegetable production in that area. Okay, it's wonderful out here in this area because it's very hot in the summer. The date palms, we say about date palms, they like to have their feet in heaven and their head in hell. Their feet want to be in good water table and their heads want to be up in mostly sunny days. They don't like rain, they don't like humidity, which is why we don't grow date palms in Florida. The humidity is too high. So this is very dry. But anyway, you can see the very close proximity of these urban areas to the ag areas. So our goal was to try and do something to keep it isolated in the urban areas and not to move into the agricultural areas. Well, do a little research and you find out very quickly that the pink hibiscus mealybug has been a pest throughout the world, Egypt, the Middle East, and in all the places where it's been a problem, there have been either natural enemies that have come in that were there originally, or that have been released there. And two of those species are Anagyrus kamali and Geranisoidea indica. Now, previous to this infestation here in the Coachella Valley, down south of here is another agricultural district called the Imperial Valley. And in the early 1990s, pink hibiscus mealybug came established down in the lower part of Imperial Valley. And there was a project that was launched of raising Anagyrus kamali and Geranisoidea indica and releasing them in that area, and basically it cleaned up the problem. So our first take on this was, rather than do a first shot with insecticides, was to see if we could release these parasitoids and get some biological control from these releases. So this is the Anagyrus kamali. It's a very beautiful parasitoid. This is one that has come out of the pink hibiscus mealybug mummy. And so then this female goes out, finds other mealybugs, lays her eggs, and the process keeps going. Well, doing a little bit more research, we needed to establish an insectary to grow these parasitoids, to grow the mealybugs. And doing some research, I learned that there was actually a nursery or an insectary for these parasitoids down in Puerto Vallarta, or just east of Puerto Vallarta, Mexico. And so somebody had to go down and figure out how they're growing these things. They're producing about 61 million parasitoids per year. Someone had to go in and figure out how to work. So I volunteered for my team to go down to Puerto Vallarta and check out the insectary. And this is actually the insectary. It's in Nayarit, state of Nayarit, Mexico. It's east of Puerto Vallarta. And they grow these parasitoids, as I said, very, very efficiently. And the process is really awesome, as they grow the pink hibiscus mealybug on squash, put the parasitoids on there. It turns out that process is very efficient, and they are able to produce, again, 61 million of these parasitoids per year. We were also labeled, and we worked very closely with the folks down at the insectary. We were also able to, they were able to help us by sending up parasitoids. This is actually one jar of parasitoids that they sent up, adult parasitoids. And we were bringing them in. We established our insectaries down in the desert. And these insectary trailers, we brought them in, set them up, one to grow the mealybugs, and one to grow 2 of the different parasitoids, Rhinosodia and Anagyrus. And in the process of that, we would grow them up. We also received parasitoids from Mexico, simply take them down and release them throughout the urban areas of the Coachella Valley. Our goal was to establish these parasitoids. And we actually, over a couple of years, released about 265,000 of these parasitoids at all of these sites, 349 sites from Palm Springs down through La Quinta and even down through Mecca in the agricultural areas down here. And to determine how well these were working, we sampled 3 of the species that the mealybug infests in the urban areas. One of these is carob tree, mulberry tree, and the silk oak tree. And we took 5-inch samples from these and brought them back to the laboratory and counted all the mealybugs on them. These are the data from 2015, 16, and 17. And the 2015, these are the average numbers of mealybugs counted on a 5-inch piece of stem. And on average, in mulberry, we were counting 500 mealybugs at this one site, 300 mealybugs at this site. On silk oak, the numbers were over 100, 150. On carob, again, 100, 150 on average. So that was in 2015, before we started the release. And as we increased the release and the parasitoids, mostly these were Anagyrus camali, parasitoids would find the mealybugs in the trees, oviposit in those trees, produce more mealybugs, and then they would go out and find more mealybugs up in the trees. And in 2016, you can see our numbers, basically non-existent. So these parasitoids really knocked the top out, knocked the mealybugs down to almost 0. 2017, there were just a few sites that had a couple of mealybugs, which is normal when you think of a classical biocontrol establishment. What was happening here is that as the parasitoids became established, they brought the mealybugs into check, and then we would create an equilibrium where you don't want to knock out all the mealybugs, because then the parasitoids have nothing to reproduce on. And so even today, I was down about 2 weeks ago, even today, there's a few of the mealybugs that are left, and the parasitoids are reproducing. So basically, we were able to knock out this mealybug population before it spilled over into the agricultural areas. Now, we still have a problem on a few of the date bunches with this mealybug, and this is what I was talking about, this mealy mass. Obviously, this bunch is not going to be harvested. But you remember those bags that I showed you that the growers put around their date bunches? Essentially, when they cover those bunches up, they essentially exclude any mealybugs from getting in. But what happens in a very, very few bunches, probably 1 in every 1,000 bunches, a mealybug crawler will get in there. These mealybug crawlers are very small. They can hitch a ride on insects. They hitch a ride on birds. They can be moved into the area by landscapers. And if one small mealybug crawler gets into a date bunch and then they cover it up, this is what will happen. So our pest management in dates now on these 1 in every 1,000 bunches, if a grower pulls the bunch off of there, sees that it has these mealybugs, put the bag back on, tie it up, lay it out in the sun, and just solarize that bunch. Don't take it out of the bag. Don't take it. Obviously, you can't take it to the packing shed. That's not going to be any good for you. The second thing that happens, so that's a cultural management practice that we tell the growers to do. The second thing that happens is the growers tell me, and I've just got a hotline established, they call me and tell me where where these infestations occur. And it really is in really spotty places in the 11,000 acres of dates that we're growing in the valley. And in the following spring, so about now, I'll take parasitoids down and I'll release them at these specific sites. And they even mark some of the trees and I'll release them. But these parasitoids are so good, they'll search for about a half a mile. So we're doing a kind of a find it. So that's our sampling. When the growers see it, mark them, tell us where they are. We're going to release parasitoids. Now, interesting little project that happened is that Sharon Andreessen, who was working in my lab, did some barcode reads and sequenced some genes of the parasitoids we were releasing. We found out that it wasn't Anagyrus camali. It was actually a different species, which we've named Anagyrus calidus. And we've done some sleuthing around and found out that sometime in those releases down in the Imperial Valley in the 1990s, there was probably some escapes of this species, which we think is actually more aggressive than Anagyrus camali. And that's the next piece of research that I want to get into. If we can actually find some Anagyrus camali someplace in the world, because all of the insects that we released in the valley here in California and all of the ones that are down there are now this new species, so it actually displaced the old Anagyrus camali. And I'd like to do some side-by-side comparisons and just look at the biology of these and see what that competitive displacement process is between this parasitoid and the old Anagyrus camali. Once again, biocontrol solution. We also need some insecticides, because the other thing that I want to do for the farmers, if they're conventional ranchers, is I want to know the trees that they actually had the mealybugs in, because when you have that big mealy mass in the bunch, crawlers can still climb up and get into the top of the crown. Those are the ones we're targeting with our natural enemies, but it also would be fine to hit the tops of those trees in the off season. And so we did some insecticides working on actually some insecticide bioassay work in the laboratory. And we have a couple of materials that we're working through IR4 channels now through EPA to see if we can get them registered for use in dates. Biocontrol solution, insecticide solution, sampling, and release solution. Now, the third pest I'm going to talk about, and I'm watching my time here, the third pest I'm going to talk about is this insect or this mite called the date mite. And you might recall this name from earlier in my presentation. The date mite is actually Oligonicus pratensis, something that I have known my whole career working in this business. It's also called the Banks grass mite, as you talked about earlier. And as I saw this pest on dates, I remembered that when I was doing my master's and Ph.D. dissertation work, I remember that this mite was described from Nathan Banks in about 1903 from date palms in the Coachella Valley of California. So I've kind of come this sort of circle here, beginning of my career, sort of ending of my career, working with this very small little thing. And that's why I said, you got to be, watch out for the small things. But let me tell you about this guy in dates. As a spider mite in a very hot region, and we know because of a lot of that work that we did with our microenvironment, that the hotter it is, the more this mite likes it. And they will completely web over a bunch of dates, like we see here. The damage that they do is they feed on the dates when they're green, and they will scarify the date. This is what a nice date should look like. You can see the scarification that's happened on these ripened fruit now. These become non-marketable for fresh market. They can grind them for paste and things like health bars and things like that, but it's not the high quality date that we want. And so when I first started working on this, guess what? They were using another dust, but this was sulfur dust. And sulfur dust is known to be very active against mites. I put this picture in, not to show you another dusting machine of these dusting, but to show you behind here, down on the ground, you see those roof lines of houses? And when I first started here, working on these date mites in the early 1990s, there weren't so many houses around the date ranches. And so using the malathion dust and the sulfur dust was more acceptable. You just kind of knew it was that time of the year if you lived in those agricultural areas. But now urbanization is coming down from that affluential area in the upper part of the valley. It's coming down, and we're having houses right on the edges of the date ranches. This kind of activity is not going to be no longer tolerated. And very quickly after this urbanization moved in, sure enough, we needed to find some different solutions rather than using dusting, sulfur dust. And besides of which, we determined that the sulfur dust the mites were resistant to it. So they were basically knocking out all the predators with the sulfur dust and letting our spider mites do the damage that they had. So one of the first things we did is some alternative miticides, and very quickly registered in 1983. We had this material, Savvy. It's now called Onager Optec, and this pretty much displaced, right off the bat, it displaced the sulfur dusting, and farmers are still using this. From 1983, we went down very clearly and said you can use it one time, early season. This material is known to have resistance in other cropping systems. We didn't want that to happen in dates. So you one shot early in the season, and it's turned out to work out pretty well to give us some time to do some other things. Well, in part of that biology, as we found out, it's not so clear in this picture looking, but this date bunch here is completely covered with spider mites. These other ones here, it's kind of a bleached out picture, but these other ones are pretty much healthy. So we noticed right off the bat that we get this spottiness of the infestations, and that the farmers wouldn't have to spray their whole field. In fact, they could actually spray the edges of their fields, saving spray, but also not inundating the whole date ranch. So it was saving them time and money. And you could also drive through and just do spot treatments of the bunches that were infested. And you could see these infestations, okay? It's not easy, but you can see them, especially in trees that are not so tall. So that was one of the first things we did. Give them a new insecticide, but say, listen, you can be smarter about how you use your miticide. You can be smarter about how you use the miticide. And so here's what they do. They go down now, you look around, and they actually can treat these bunches. There's a guy on a tractor treating on a tractor, and they've got high booms to treat the taller trees. So new miticide, better way to use it. Now, we also notice that there are a number of predators that are out in the date ranch. The six-spotted thrips is a nice one. There's a spider mite destroyer, which is a coccinellid, a minute pirate bug, aureus, and then there are a number of predatory mite species in the family Phytocyidae. So in the last, since the early 1990s to the present, I've been working with biocontrol, and one that we've worked a lot with is this phytocyid mite, Gallindromus fluminus. And we know that it survives high temperatures, which is not usual for a phytocyid mite. It prefers the eggs and larvae, so we need to release it early. It's not impacted by the Savvy, and you can't put out too many of them because they don't like each other. And if you put out too many of them, they'll start eating each other. So armed with that, we went to the field and did some trials and found some preliminary results that looked pretty good. And then our studies got disrupted by a predatory thrips, which I'll talk about in a second. But I do have a company that's looking at producing these things for releasing in the date ranches down the valley. Here's that thrips that I was talking about, and this picture was actually taken in one of our studies where we were studying the predatory mite. Put the predatory mite out, everything was going fine, and then this thrips came in and ate all the spider mites and all the predator mites. So it got me thinking, well, maybe we need to be doing some work on this. And so in addition to the predator mites, started working on the predator thrips. And just this year, or last year, we had one of our farmers that found a supplier for these predatory thrips. And the way that this grower received these thrips is that they were grown in flats of bean. So the way that these are produced is flats of bean are planted. The bean plants grow up in these flats, and these flats are probably 14 inches by about 18 inches. grow the beans up, they infest them with spider mites, let the spider mites develop on the beans, and then they put the thrips in there. And while the thrips are still in the nymphal stage, you can take these bean plants and you can cut them down and literally put strands of these bean plants in over the crooks of trees. They use these in almonds and other crops. And we actually had a farmer who tried this last summer in his date bunches. So this is one of those strands of the bean plant that had spider mites, different species of spider mite now, but also these thrips. Put them into the date bunch, and they were treated the week of June 14th. I found out about it a week later and asked the grower if we could come out and sample. He said, absolutely, come out and do what you want on the ranch. He treated it when the infestation was a little bit high, about 13%. And that's important when we talk about the preliminary results that came from this trial. Well, these were tall trees. And here I am up on one of the lifts. The grower brought his lift out, let us go up into the trees so we could sample these bunches. And we sampled these on 3 different times. Here we are sampling. This is Shayla Hampel, one of my technicians. And here I am sampling. So we're looking for spider mites and we're looking for thrips up in these bunches. And again, preliminary data, but what we found out that there were more thrips in the treated bunches on our first sample date. On our second sample date, though, the spider mites had actually grown and done a lot of damage there. But then by this time, we were kind of late in the season. By this time, the mite population had started to decline. That happens naturally in dates when the fruits start to ripen and the skin gets a little bit, it starts to turn color, not so green. The spider mites will go away. They don't like to probe through, and the skin gets a little thicker and it gets a little sweeter. And that's one of the things I remember from my research in Texas, is that these mites do not like high carbohydrates, which is what we were looking at in the sorghum. So it makes sense when the fruit starts to ripen and put on more sugars that you would have the spider mites would not be on those. Anyway, in our second sample date, the treated bunches didn't have much on them. The non-treated, these are just averages. We don't even have statistics, enough data to draw statistics on this. But then back here on the third sample date, there were not any thrips at all. More importantly, though, when we look at the infestation, this line right here is the non-treated, and you can see the infestation got up here to about 52%. On the treated ones, even though we treated at 13% infestation grew, and then it kind of leveled off. So at least preliminarily, it's giving us some ideas that these thrips had some impact out there. And so we're hoping that moving forward into this next year, I'm going to do some more controlled studies where we bring these strips in and do that work. Now, finally, I'm going to finish up real quickly here because I know I've got a little bit long. Once again, we're working with these spider mites in these very tall trees, and it's kind of hard to find them. So we've moved, like most people in agriculture, to some drone technology, mounting cameras, and working with this fellow. He's got a private business. His name is Terrence Powell. And Terrence has mounted some cameras, infrared cameras, to go up and take a look. This is a visual image of spider mites, and then he's using infrared photography to look at these hot spots. And when the mites feed on the photosynthate, then the fruits stop photosynthesizing and they become warmer. And you can detect that with IR photography. And in this particular study, Terrence's drone and the algorithms that he wrote to recognize this, the actual infestation was about 4%. His red correspondence was about 6%. And so it's kind of fun playing with Terrence and his drones to do some sampling for this. And the other thing is he's looking at is some predator release technologies, because again, some of these tall trees, we want to be able to drop these predator mites and predator thrips maybe onto the tops of the date palms. And so we're working on that strategy. So I'm just going to finish up, and I chose this picture because depending on if I'm going to the valley, which I head east, this will be the sunrise, or when I'm coming home from the valley and I'm going west, and this will be the sunset. For me, this kind of represents this sort of full circle of working with this little spider mite, this Banks grass mite, from the time that I started to the time that I'm basically finishing my career. And I had no idea when I started out in this business that I would be working on such a small insect, on such a small mite, on such a diversity of crops. But what I've tried to do is show you my philosophy about integrated pest management is I want to provide tools to the farmer. Farmers are going to choose whatever they are comfortable with choosing. If they're conventional, they're probably going to choose sprays first. Let's give them better sprays. Let's teach them how to use the sprays. Let's teach them how to sample. Let's give them those tools. Organic farmers want things like mating disruption, classical biocontrol, augmentative biocontrol. And that's what I've done over my career, is to try and provide farmers choices of pest management tools that they can use. And so with that, I've got a lot of people to thank, but I'm not going to go through this. You'll know very few of them anyway. But I've had a lot of folks working on this with me over the years, and lots of good cooperators and good funding sources. And at that, I think I'll just stop and take any questions.
Hey, Tom, this is Jim Kalish.
Hey, buddy.
Hey, how's it
All right.
Hey. I was noticing on some of the images and the various studies you were doing that the ground litter was variable in those home ranches. Some of them were very clean and some of them were cleared. I mean, how much does sanitation pay or impact the presence of some of these pests like the carib moth and the like?
Yeah, it's huge. It's huge. The first question I asked, because those of us who actually worked on corn and sorghum and things that are on the ground, yeah, there are a lot of grasses, and as we know, Bermuda grass and all these grasses are great hosts for the Banks grass mite. And the farmers that keep them clean don't have this bad of problems, but it doesn't solve the whole problem. Farmers that have really dirty ranches, it can really be a problem. One thing you don't want to do, though, is if you have a lot of weeds in your ranch, is you don't want to go through and disc those out during the season, because the dirt rises. As you know, Jim, these mites balloon, they blow on breezes, and it takes one pregnant female to get up in a date bunch. Once they get up there, these mites double their population in those temperatures of the valley every 36 hours. So we get these explosive things. So very good question. I work with the growers to keep the ranches sanitized, but still, mites can blow in from a neighboring crop, and there's a lot of different vegetable crops there. Great question. Tom Holzer. Oh, man. You're muted. You're muted. You're muted. You're still muted. I love it.
Unmute.
There
I got
you
it.
go.
Did Hey,
I realized looking at my image right next to yours, Tom, that I'm starting to look a lot more like you as I get older.
that thought struck me as I see you sitting there talking. Boy, that's amazing. I don't know which one of us should feel the worst.
Neither one. Just is.
It
Rhonda,
sure is good to see you.
nice
I'm
to
sorry I
see you.
missed your talk. Apparently everybody liked it.
Well, I think it was recorded, so maybe we can. There's Jim. Good to see you. Jeff, Rhonda, man, Sripat. Great. Andre, how we doing? Oh, there's the classroom. I can't tell who's there, but probably a classroom that I sat in one
We have
time.
all sorts of people here. We have Dr. John Rusin and Gabby and Tasha, Mika, Rogan, Nina. Oh, look at that.
Oh, check this out. Check Jeff Bradshaw's got his poster up. The stridulators,
Oh, yes.
2010. Oh man.
Oh,
That was, yeah, that was the time. Let's get the band back together.
Does
Oh,
anyone
man.
else have questions? I mean,
Yes.
it's okay, keep reminiscing, I don't wanna cut that off. I'm also gonna try to not get shaky here, but, yeah, if there's any other questions.
I've got a question, Tom. This is John Riverson.
Hey, John.
Tom, you've done a lot of great work with a lot of different critters, and dealing with the invasives is a double challenge because you're having to deal with identifications and get that critical taxonomic information in place.
Yeah.
From your perspective, where do you think we are right now with our taxonomic expertise to address those kinds of issues, since they're not going to be getting any easier in the years to come?
Yeah, no, I mean, compared to when I was taking taxonomy at University of Nebraska-Lincoln, our molecular tools are so powerful now, and so pretty straightforward and pretty easy that you can get pretty quick identification. I never made the real leap into molecular biology, but we have some folks here in our department that are very quick at identifying different species. And many of these invasives are tiny. They're mites or they're small thrips, and we've gotten very good at. Very first thing you got to do is figure out what it is, and make sure that it's not something that is already here. I think, I think that it's much more advanced. Could we use more? Absolutely. So does that help? Does that kind of fit with kind of what you're seeing, John? What you think?
Yeah, I'm just kind of curious. Looking at we've got some great tools. I'm just wondering about what's going to happen with the people who actually use those tools, and having those systematists out there who can help us actually interpret that and put it into an appropriate evolutionary and systematic framework.
Yeah. No, I understand your question a little bit better now. Yeah. I mean, when you're in this business for a long time, you see the ups and downs and you see the swings and when I graduated statistics, you had to be really super strong in statistics, right? That was the number one thing. And then kind of pendulum swung and we kind of moved a little bit more away from that and then moved into, you've got to be a molecular biologist. And we have to have both. I mean, what I've trained my students now who are IPM students is you've got to have some understanding of molecular biology so you can at least understand the tools, whether you use those tools or not. So I think the strongest students are being trained in both, being good taxonomists, as well as having good understanding of systematics and at least a basic understanding of molecular tools that you can use for identification. These tools are not very difficult anymore. So I think you got to have both. And what I've seen with departments is these swinging things. And our department has done the same thing. We've lost a lot of our IPM kind of field applied entomologists, and now we're going to swing back and look at hiring when we can actually hire again, hiring replacements for some of us who have retired and are getting close to retirement.
Thanks, Tom.
Anyone else have any questions? Awesome. Well, thank you so much, Dr. Perry, for speaking, and again, congratulations. And yeah, we're going to be sending out the recording to definitely our department, and I'll send that to you as well if you want to distribute that.
Love it.
Yeah,
that'd be great. That would be great. Well, it's been a real pleasure, and it's truly an honor to be selected. I again, like I said, I have really fond days of setting in Bruner Club meetings and making decisions about Wiedemann Trust Awards and all those sorts of things when I was there, and a really fun time. Tom, I told them that our office was up in the northeast corner of Plant Industries at that time, and it was really cold up there in that building with Kim. And, making our tea and trying to stay warm. Rhonda was up in there. Jim, you were up there. So it's really, really fun memories. And I've only been back to Lincoln one time for a wedding, but I'd like to correct that and maybe come back and visit sometime. If there's no other questions about any presentation or anything else, anybody who'd like to just stay on or just chat about how life is, I would love that. Because I see some really good old friends, good friends there.